If you want to learn, this is an extra information actually. If you want to learn the structures of proteins and other macromolecules, we have got a site that is where we can obtain the structures of protein, and this is a bank actually, therefore it is called as a protein data bank. These structures are available freely in the public domain. You need not to pay anything. You just put in the Google protein data bank, you will get this site, and you just write name of a protein over here. So in our case, now we are going to learn the structure of alpha glucosidase. So what we will do is we will search here alpha glucosidase. Already done it, and then you will get window like this. So this is a unique code. Like we have EC numbers for enzymes. Here we have PDB IDs for uh, proteins. So this is co-crystal structure. If I click, this is actually alpha glucosidase. If we try to observe it in 3D form, this is how protein will look like in its 3D form, and this is how we can rotate. This is actually a three-dimensional structure of enzyme alpha glucosidase, and it is in complex with drug alpha glucosidase in complex with Vogelibose. So we'll see how Vogelibose remain in bound form with alpha glucosidase. So this is actually X-ray diffraction structure, and here you will find a crystal structure of alpha glucosidase with Vogelibose. So I'll click over here. Here I've got the structure. This is a structure of an alpha glucosidase enzyme, and here this is sequence which has been analyzed already by scientists. Now here you will be able to see structure of Vogelibose is there, and it is there in complex with uh, alpha glucosidase, and here it is written ligand interaction. So in order to study how ligand Vogelibose become interacted, or how it become bound to alpha glucosidase, we'll see that. So I'll zoom out in order to observe it very clearly. So this is 3D structure. This is how we can rotate anywhere the way we want, and then we'll be able to see structure of a ligand which is there. I'll try to show you interaction, that is a different kind of interaction which occurs between drug and enzyme, or in general protein, in two-dimensional form, so that it will become easier for you to understand. So if you observe carefully, as enzymes are complex structure, they are generally protein in the nature. So this protein is made up of a sequence which is there in front of you. Now if I try to highlight this, if I click over here, you will be able to see portion in the red, that is polymer portion. This is a ligand portion which is there at the center, which has become pink in color. And surrounding means, if you observe now carefully, surrounding means it is a catalytic site of this protein. I'll zoom in. Now you will be able to see in very correct manner. So this portion which has become pink, now this is a catalytic site where our drug Vogelibose is there, which has become linked, and some of the amino acid residues are there, and even you will be able to see some water molecules if it is there in shade color. Just recall structure of Vogelibose. I'm marking with pink color. Now this is the one which is there in the ligand binding domain or catalytic site of alpha glucosidase. So this is portion, that is molecule, is Vogelibose, which is there in pink color. When I'm touching cursor to a molecule, it becomes pink, and somehow a dotted line is indicating how interactions or bonding is there between different functional groups of Vogelibose and amino acid residue in present in the catalytic site of the enzyme alpha glucosidase. So This is how we can learn how particular mode of action, or we get idea about a fair idea how drug binds to their targets. I'm deleting polymer now. That is, polymer means in our case it is our enzyme. So now only drug glucose there in front of you. This is a editing feature. Like we can hide any particular portion or any particular compound, or sometimes even protein. This is our future of software. Mind well, no one is going to ask you how proteins and whatever I have shown now. No one is going to ask you in the exam, in the viva or something like that. This is just an inside how drug binds with their proteins or enzymes or any other suitable target. Don't bother about it. Just for sake of simplicity and understanding, I have made mention about this.